Thank you. Let me start with a couple of disclosures. So first of all, this is a CME conference, and I regret that I have no conflicts. Second, that I'm going to talk to you about a new topic for me, cancer biology. And I know little about this. I appreciate if the audience has comments, and particularly if I say anything particularly egregious or stupid, corrections. Those would be well appreciated. And the third is that the data I'm going to show you, some of which is quite elegant morphology, was generated by a research scientist at Edinburgh Hospital, Cambridge University, Rafia Al-Lamki. She deserves credit for the quality of the data. The misinterpretations that may arise from this, you can attribute to John, Wang, and myself. So by way of introduction, I got into this particular issue of TNF and renal cell carcinoma by a longstanding interest in tumor necrosis factor, TNF. TNF is now regarded as the prototypic inflammatory cytokine. The name, related to the fact that it has something to do with tumors and may cause the necrosis of tumors, really was given by Elizabeth Cardwell and Lloyd Old in a 1975 paper in which they described the fact that LPS-stimulated macrophages secreted some molecule that was capable, when LPS was removed, of causing the hemorrhagic necrosis of tumors. It's now very clear that although there are some tumor cells that can be killed directly by TNF, the principal anti-tumor activities of this molecule are caused by the induction of changes that promote thrombosis and inflammation in the vascular endothelial cells. And tumor endothelial cells, for reasons that are not entirely clear, are much more susceptible to these actions of TNF than are the endothelial cells that line the rest of the vasculature. The last thing I'll tell you is that the responses to TNF in vitro, but particularly in vivo, are quite complex, often contradictory. Many papers will report that TNF does one thing, and then several papers will report it does exactly the opposite. And some of this contradiction and complexity arises because there are two distinct TNF receptors, known as TNFR1 and TNFR2, and that they are independently regulated and they mediate very different kinds of responses in tissues. So the story I'm about to tell you began with work that came out of a collaboration of John Bradley, who now is the research director at the Cambridge University Hospitals, and myself. And we were interested in understanding some of the signaling pathways, and particularly some of the questions associated with these two different TNF receptors. And the slide that I'm projecting is some work that John generated when he first left Yale and went to Cambridge, using this new prototype instrument that Brad Amos had invented called the confocal microscope. And what John was able to show is that if one looked for the TNF receptor 2, it was expressed, shown here by the green fluorescence, superimposed on a phase image, as basically a cell surface protein diffusely over the cell surface. But TNFR1 was localized to this structure that is perinuclear and turns out to be the Golgi apparatus. Now, we had shown that TNFR1 is involved in the characteristic pro-inflammatory changes as well as some of the apoptotic responses that are mediated in response to this cytokine. It activates the I-kappa B kinase pathway, leading to the activation of NF-kappa B. Wang Min showed that it was coupled to stress-activated protein kinases through ASK1, activates JNK, and then it's AP1, and it's also known to activate caspases, particularly caspase 8, and in humans also caspase 10. And all of these responses are mediated by different signaling complexes that change over time and move to different portions of the cell. TNFR2 was a bit of a mystery. If you overexpress it, you'll activate NF-kappa B, but ligand binding to this receptor in our hands did not do so. And we could never figure out what TNFR2 was doing. Wang made the observations that in endothelial cells, at least, it was coupled to a cytosolic tyrosine kinase called ETK, which in turn would phosphorylate some of the tyrosine residues in the cytoplasmic portion of the VEGF-R2 receptor, and then independent of VEGF, activate a PI3K AKT pathway. Now, we have studied TNF receptor expression in normal tissues, moving from cells, and that's what got us into kidney. John Bradley is a nephrologist and ran the nephrology section at Addenbrooke's Hospital. And this work from an early study documented that TNFR1 is expressed in kidney, but you can tell from the red staining, not uniformly, and in fact, it's almost entirely restricted to the endothelial cells of the glomerulus, but also occasionally to endothelial cells in the microvessels, peritubular capillaries. And no other cell in the kidney seems to express any significant amount of TNFR1 in what we regard as healthy tissue. On the other hand, TNFR2 was virtually absent from normal kidneys. By immuno-EM, we could find a couple of gold particles on some of the tubular epithelial cells, but by immunoperoxidase staining or immunofluorescence, it is virtually absent in normal kidney. On the other hand, these things changed quite radically when we started looking at biopsies that showed renal injury, many of these from transplant recipients, but some of which also came from ischemic kidneys. And basically what we saw is that TNFR1 fundamentally disappeared from the endothelial cells, and it was replaced on the endothelial cells by TNFR2, but TNFR2 was also expressed at very high levels on the tubular epithelial cells. And by in situ hybridization, we're able to show that the TNFR2 was probably being synthesized by these cells rather than simply being extracted from the urine. So there is a change in the nature of the receptors. Now, to try and understand what these receptors were doing in kidney tissue, we developed a model of renal organ culture. We would take pieces of human kidney that we typically got from resections, nephrectomies, that were done for removing renal cell carcinoma, and which tends to be localized often to one pole of the kidney. And when the rest of the kidney is removed, you basically have normal tissue. And so we could set up from these experiments organ culture of these pieces of normal kidney and then ask what would happen if we treated them with TNF. But we also had this trick that my colleague Walter Fears had described, the fact that you could mutate TNF to produce so-called mutants that would selectively signal through one receptor or the other, as opposed to wild-type TNF, which signals through both receptors and binds to both receptors with approximately equal affinity. And then we could assay the responses of the kidney tissue by histochemical or immunohistochemical assays. So this is a summary of some of the data that we had collected, that if one uses tunnel staining to look at apoptosis, for example, we could find that wild-type TNF induced apoptosis in endothelial cells and in tubular epithelial cells, and that the apoptosis was principally mediated by TNFR1, shown by using the mutation that only binds to that receptor, and there was little apoptosis when we used the mutation that was selective for TNFR2. On the other hand, if we asked about proliferation, and we did this by a variety of techniques, nuclear staining for PCNA is shown here, one could see that, again, wild-type TNF caused a significant amount of proliferation, and it's the R2 receptor triggered by its mutation which leads to the proliferation of these renal cells. So that was the background that got us into this, trying to understand the inflammatory and reparative responses of TNF on normal kidney. But John Bradley had the idea that maybe, given the fact that we were seeing a proliferative response, we ought to be looking at renal cell carcinoma. Now, about 75% of renal cell carcinomas are malignancies of the tubular epithelial cells of the clear cell type, and most of these tumors have inactivating mutations of the von Hippel-Lindau protein. Now, the von Hippel-Lindau protein normally serves to target HIF1 alpha or HIF2 alpha for degradation, and when it is non-functional, HIFs are elevated. The HIF protein forms with HIF1 beta, a functional transcription factor, and you produce a lot of target genes, including vascular endothelial growth factor, and these tumors tend to produce a lot of VEGF, and they also express VEGF-R2 receptor and will respond in an autocrine fashion. And anti-VEGF therapy, either with antibody or targeting the kinase activity of the receptor, has some efficacy, and it's an approved agent for use in renal cell carcinoma. TNF is also elevated. You can find it in the tissue, you can find it in the blood. The relationship between VHL and TNF is a little less clear, but there's at least one report that suggests that VHL normally interferes with the translation of TNF message, and that when VHL is inactivated, you increase translation and you increase the production of tumor necrosis factor. It's a little less clear what role TNF plays, but there have been a couple of phase 2 trials in England using anti-TNF antibody that have shown about 30% response rate in renal cell carcinoma, either partial or complete remission, suggesting that it too might be an autocrine growth factor for these tumor cells. So the study I'm about to show you has been an analysis of clear cell renal cell carcinoma, looking at the expression of TNF, TNF receptors, and the effects of TNF on this tissue. So this is simply an example to show that these tissues express, the tumor expresses very little TNF-R1. You can still see it on normal glomerulus or on peritubular vessels, but there's hardly any TNF-R1 on the tumor cells. On the other hand, the tumor cells express TNF-R2. And if one categorizes tumors according to their malignant grade, the Foreman grade, there is a progressive increase in the expression of TNF-R2 with increasing signs of, or evidence of increasing malignancy of the tumor. So TNF-R2 is expressed by the tumor cells as opposed to TNF-R1. Now, this is simply an organ culture experiment of a control piece of renal tissue that contains renal cell carcinoma. And one can see that TNF-R1 is expressed in this tissue mostly on vascular endothelial cells, not on the tumor itself. And this is a signaling molecule, ASK1. Wang Min described the fact that when TNF-R1 is engaged, you convert this receptor from the inactive form, which is phosphorylated on serine residue 967, to the active form, which loses this phosphate group but now becomes phosphorylated on threonine 845. And the point is that there is hardly any active ASK1 in the basal tissue, even though the receptor is expressed and the enzyme is present. On the other hand, TNF-R2 is brightly expressed not only on endothelial cells but also on tumor cells. And on the tumor cells, one can find phosphorylation of ETK, the endothelial tyrosine kinase, that's downstream signaling molecule. And if one takes these organ cultures and treats them with the R2 mutein, you don't do anything to TNF-R1 levels, you don't activate ASK1, but you increase the expression of TNF-R2 and you activate the phosphorylation of ETK. And not only do you activate the phosphorylation of ETK, but you also activate the phosphorylation of VEGF receptor 2. That in the absence of VEGF, this receptor is now becoming activated in response to a TNF-R2 signal. So this is in a renal cell carcinoma cell in situ, in an organ culture, the same pathway that Wang had described in cultured endothelial cells. So this is a summary of the data that Rafia generated, looking at the responses. If we look quickly, what you can see is that TNF-R2 mutein increases the level of expression of TNF-R2, increases the phosphorylation of ETK, activating it, and increases the phosphorylation of VEGF-R2, activating the kinase activity, we believe, of this enzyme. TNF-R1 mutein does none of these things. It increases TNF-R1 and activates ASK1. And wild-type TNF does both of these things. So the natural molecule is engaging both receptors, is triggering both responses, but the downstream responses appear to be quite different. So this is an experiment to see what happens in terms of cell death. So the renal cell carcinoma has a certain degree of apoptosis going on, activated caspase-3, some tunnel positivity. It's not affected by the R2 binding mutein. This does not activate any significant apoptotic response. On the other hand, the R1 mutein and wild-type TNF, as you can see, activates caspase-3 quite dramatically and also increases tunnel staining, evidence that cells are degrading DNA and dying. And one can summarize these data and show that in the tumor cells, R1 mutant or wild-type TNF induces quite a bit of apoptosis. R2 mutant does not. Similarly, one can ask about entry into cell cycle. And this is done in this case by staining for phosphorylated histone 3. We've done similar things with PCNA and other markers. But the basic point is that wild-type TNF and the R2 mutant increase the number of nuclei that are in cell cycle. R1 mutant does not, similar to untreated cells, and these are the quantitative data, that there is a proliferative response of the tumor cells mediated by TNF-R2. So let me summarize what I've told you, where the story stands at the moment. We know that TNF-R2 is upregulated not only on injured renal epithelial cells, but also on malignant renal tubular epithelial cells in tumors of clear cell type. We know that engagement of TNF-R2 on these cells activates ETK, as we can see in our organ culture experiments, and appears to transactivate VEGF-R2. It also induces entry into cell cycle, and we don't know whether this is mediated by VEGF-R2. Those experiments are in progress to see if a kinase inhibitor will in fact block the TNF-R2 response. We know that these cells make TNF, the tumor cells do, perhaps because of the inactivation of VHL, and that TNF can act as an autocrine growth factor, and that neutralizing TNF with an antibody has had some efficacy in clinical trials. But the point we'd like to make is that you are, in a way, limiting your efficacy if you neutralize TNF, because you not only block the growth-promoting activity of this mediated by TNF-R2, but you also are blocking the apoptotic effect on the tumor cells mediated by TNF-R1, as well as the classic anti-tumor response of hemorrhagic necrosis mediated by TNF-R1 on the endothelial cells that are feeding the tumor. So the point of which is that we think that there is much better use for an inhibitor, if one could be developed, that would specifically target receptor 2, spare the actions on receptor 1 instead of targeting the ligand, and that perhaps this is mediated by VEGF receptor 2. So a kinase selective inhibitor might be effective, and that those are experiments that are in progress. And the TNF-R2 and its downstream effectors may be a therapeutic target in renal cell carcinoma. So as I told you, these data were generated by Rafael Almanki. John and Wang and myself were involved in the design and conception of these experiments. And there's a bunch of people at Cambridge and Edinburgh's Hospital in John's lab and in the tumor group there who helped in the analysis and collecting tissues for us. And I'll stop there. Thank you.